Good morning. Welcome again to this presentation about immune system. We will talk today about antigen presentation to lymphocytes, but before that, I would like to wish you Happy New Year, full of health and happiness, and we hope that will be much better than the previous one. The story has started in around 1952, when Jean Dosset, head of the blood transfusion center in Paris, has observed some incident during blood transfusion performed in polytransfused subjects. He realized, as well as his technicians, that the blood vials used were rejected, or the blood was rejected, although the blood vials used were compatible with the blood system known until then. So something has happened, and kind of rejection, not allergy, but rejection of the blood. And so he designed this slide here where the donor, the blood of the donor was rejected by the receiver, and there was agglutination. So the recipient serum contains antibodies that agglutinate the donor's white blood cells. At this time also, in the 60s, attempts to transplantation were also unsuccessful. And in this very pessimistic context, that thanks to the discovery of Jean Dosset in 1952 of the tissue groups, it was realized that the graft transplantation required that the donor and the recipient be histocompatible, and that the tissue groups became the major histocompatibility complex, or MHC. So the concept was born after this incident, and around the 70s, it appeared that MHC molecules were not only used to reject transplant, but played a major role in immune responses. The MHC became HLA system, a human leukocyte antigen, a set of antigens expressed on the cells of organisms, markers of the identity of each of us, markers of the self. Indeed, epidemiologists were always wondering why in some pandemics, like the actual one or Ebola pandemic, there are always survivors, are always people they can manage to get through, and they will not have any symptoms or just mild. And that because one of the explanation is they have this MHC molecules, these genes, that can allow them to fight against new diseases, even their parents or their grandparents didn't know about these diseases. So the concept of polymorphism was born. Most of the genes that exist in a given species are unique. By expressing themselves, they are translated into a protein which would be found identical in all individuals of the species considered. These genes are called monomorphic genes, which are the normal common genes. Some genes can exist in several forms called alleles. Those in the ABO system of blood groups, the responsible gene exists in two allelic forms, A and B. These genes are weakly polymorphic. We have A, B, and O. The HLA system has a very high degree of polymorphism, i.e., for given genes, many alleles are possible. We can assume that we have about 500 alleles from the mother and 500 from the father. So you can multiply, and you will see that we have about 250,000 possibilities to have a gene. That's why we are unique, except of course of true siblings. Multi-allelic genetic polymorphism. In the human species, there are a very large number of alleles for each HLA gene, several hundred for the most part. That are the most polymorphic genes in human species. Each individual is heterozygous for the most of their HLA class 1 and class 2 genes, and express only one of the two of the alleles of each gene present in the human species. This characteristic makes us, or each individual, almost unique. So basically, we will become genes, especially HLA system or MHC, or animals also, will receive them from parents, and of course we will express some of them. And that was why we are almost unique in terms of expression. APC. Antigen-presenting cells, or APC, capture the antigen, conditions them, and present them in immunogenic form to lymphocyte. That's why the topic was called Capture of the Antigen and Presentation to Lymphocyte. Who is capturing antigen-presenting cells? Who is receiving the lymphocyte? Who is doing the job between are the MHC class molecules. But keep in mind that there is always in immunogenic form, which means that these cells that are capturing the antigen, for example, microbes or viruses, are not presenting any protein. They are presenting mainly the immunogenic form that will trigger the immune reaction. MHC, major histocompatibility complex molecules, are expressed on the surface of cells and participate in the presentation of antigen, which means each cell, either nucleated or somatic cell or professional cell, will present these MHC molecules. We will see MHC class 1 versus MHC class 2, and what kind of T cells. So always we have a cell presenting MHC molecules, we have a lymphocyte, and between them we have MHC class molecule, and that's why we will see that we have T cell, CD4 and CD8 based on MHC, and the cell is presented. There are two classes of MHC. Class 1 molecules are present on the surface of all nucleated cells in the body, while MHC class 2 molecules are expressed only on the surface of antigen-presenting cells. So every cell which has nuclei, nucleated cell, expresses MHC class 1. Only now professional cells, macrophages, dendritic cells, B cells, can express MHC class 2. And then we will see what kind of T cells will respond to that. Receptor for antigen. Receptor for antigen are fixed in the membrane of lymphocytes. Thanks to this receptor, each lymphocyte specifically recognizes an antigen. The receptor for T cell antigen is a molecule called TCR, T cell receptor, and is associated with the CD3 surface molecule. The receptor for the B cell antigen is a membrane immunoglobulin. We will see here that all this, here we have CD4 cells, and we have CD8 cells, and we have, of course, here MHC class 1, and 2. So MHC class 1 are present in all nucleated cells, and they are presented to CD8 cells. MHC class 2 only on the dendritic cells, macrophages, lymphocytes, and some endothelial cells. They are presented to CD4 cells. So MHC class 1, CD8 cells, MHC class 2, CD4 cells. Which means, just keep in mind, I will always give this advice to my student, the multiplication of the MHC with the CD4, with the CD will give us always 8. MHC class 1, multiply 8, it's 8. MHC class 2, multiply 4, it's 8. If you do a mistake, MHC class 1, multiply 4, it's 4, wrong. MHC class 2, multiply 8, it's 16, it's wrong. So MHC 1, CD8, MHC 2, CD. Question. How do few rare lymphocytes find a given antigen that enters the body from anywhere in the body? Indeed, the body is huge, it's extremely big compared to the tiny measurement of microbes. In the micro or nano viruses, you cannot see them even with a normal microscope. And then the entries of the body are extremely diversified. The second question, how is the immune system organized to achieve an appropriate response, i.e. antibody against extracellular microbes and cytotoxic T cells against infected cells that contain microbes in their cytoplasm? Entry of the body. The microbes can enter the body through different ways. Through a skin that did not, like here, did not heal, contact, through when we sneeze or cough, 100,000 of germs can be ejected, rejected, and of course projected to the face of the person in front of us, and of course through bites, of course, and we have also the digestive system. So basically, we have variable entries and tiny germs with this huge body. In contrast, we should have a immune system that allows us to always try to attack, neutralize these microbes before they can hit the appropriate organ. For example, the digestive system, which we would call the intestinal barrier, it's the size of this tennis court, for example. The mucus itself can measure, it's huge. The mucosa is about 300 to 1,000 square meters. The microbiota about 10 to 14 bacteria, which is more than we have cells in our body. So basically, we are more bacteria than humans, if you can say, compared to the number of cells of bacteria. And we have these huge surfaces. How we can now control all these germs that can enter the body from anywhere? One of the tools to control are what we call them dendritic cells. If you see them, they are beneath any epithelium, the skin, the respiratory tract, as well as the digestive tract. And these dendritic cells are always here. They will capture, we call them also Langerhans cells. They capture the microbe, phagocyte, and present. That's why we call them antigen presenting cells, capturing, phagocytosis, and presentation. Once they have captured, they will become mobile. You see them? They will move through the afferent lymphatic vessel to the nearest lymph node. Why they do this move? Because they will have to present it with the name antigen presenting cells to the T cells. T cells, we can find them in any lymph node. And here you see all those stations between the contact of dendritic cells with the germ and lymphocyte are all over the body, everywhere. Even we have also the spleen and also the gut, what we call Peyer's patches, spleen and the gut. And of course, we have here the tonsils, which is the entry of the respiratory tract, and the Peyer's patches are the entry of the digestive system. Basically, we have all those stations. We call them stations because they will increase the possibility between the contact of antigen-presenting cells and the lymphocytes in order to present what they have captured. Here we have the histological section, and here, of course, we have here just a schematic section, and you can see that the lymph node is well organized. There is the cortical part. It's reserved or occupied by B cells. Paracortical part or paracortex is occupied by T cells. So in the periphery, we have B cells, and almost in the centrum, we have T cells. Why do we have B and T cells in the lymph node? When the dendritic cells are capturing a germ, they will go through this lymphatic vessel and then channel, and they will present to T cells, naive T cells. But assuming that the germ was directly injected to the blood through a bite of a mosquito. In this case, dendritic cells cannot capture. So basically, the germ is free, either in the blood or in the lymph. So basically, in this case, the germ will be captured because it is free by B cells. I will repeat again. B cells, they can capture the free germs that are circulating in our system. And T cells, they will be receiving the germ through antigen-presenting cells. The antigen-presenting cells can communicate only with T cells, while B cells can themselves capture the germ, and then we will see how the communication will occur. So the lymph node, as well as the spleen, as well as the tonsils, as well as the Peyer's patches, are stations where we can increase the possibility of presentation of the germs, even if there are few, to the T and B cells, either T through antigen-presenting cells or B with free germs in the blood and the lymph. The same thing for the spleen. As you see here, we have B cell zone as well as we have a T cell zone, and then there is a communication between the two, of course. When a germ is captured, as you see here, through the epithelium by the antigen-presenting cells, it will move through the lymphatic vessel and then go to the T cell and present it. Now, if the germ is free, especially in the blood, it will move to spleen, and then in spleen we have blood-borne antigen are captured by antigen presented in the spleen. And lymph node captures antigen from the epithelium and the connective tissue. So basically we have ways and organs, they will help us concentrate in the communication between the germ and the cells, either T or B. B and T cell response. Do the B and T respond to the same way, to the same antigen? No. B and T differ in the type of antigen they recognize. B cells, they can recognize a wide variety of molecules, big, such as proteins, polysaccharide, lipids, nucleic acid, and chemicals. Repeat again. B cells can recognize a wide variety of macromolecules, like proteins, polysaccharide, huge molecules, complex, lipid, and acid, nucleic acid, as well as chemicals. And how can they do that? Because they are also, they enter under the category also of antigen-presenting cells. B cells, yes, they produce antibody, but they have the capacity to phagocyte, recognize, phagocyte, and present. That's why we said B cells are among also the antigen-presenting cells. But they can recognize the germ in nature or native form, rather than the T cells, we would see some restriction. Now, the immune response to B cell can be generated by different types of microbial walls and soluble antigen. T cell now. We have a T cell here, T cell receptor, and we have, of course, the antigen-presenting cells and MHC class molecule. Now, what do they recognize, T cells? You see too many yellow things. T cell can, one restriction, only recognize fragment of a peptide protein. Not even the protein. Fragment of a peptide, not even the whole peptide. And second restriction, only when these peptides are presented by specialized cells. Second, T cells are not capable of phagocytosis. T cells, they cannot go to the germ and phagocyte the germ. No. T cells will wait until another cell will do the job. Macrophage, antigen-presenting cells, dendritic cells, B cell, they will phagocyte. They will, of course, destroy the germ and then present the immunogenic part to the T cells. So two restrictions, only fragment and only when there is presentation. We have seen earlier, B cell can recognize proteins, polysaccharide, nucleic acid, chemicals. Now here we are talking just about amino acid. And usually about 10 to 20 amino acids, not more. Otherwise, they cannot recognize it. T cell response is associated only with antigen protein, which are associated with the host cell. The induction of the immune response by antigens is a remarkable reaction to bypass the virus barrier. So we have barriers, not physical, but we have challenges. The first one, the reduced number of naive lymphocytes and differentiated. We don't have billions, trillions, unlimited T cells. We will see it in the next lecture that T cells are differentiated and they are born, of course, in the bone marrow, differentiated in the thymus, but we have just a limited number because the thymus at puberty, after that, it doesn't work properly or correctly to produce more T cells. So we have limited number. And immunologists came up with an ideal number. Sorry, this is 10 to the 5th. They said for every contact with an antigen, through, of course, antigen-presenting cells, the immune system will mobilize one T cell out of 100,000. For example, you take a baby, for example, to vaccination, or a baby is exposed to a germ enters, for example. If we have an antigen and the baby has 1 million T cells, just numbers, of course, just to make it clear, the immune system will mobilize 10 T cells. The rest will remain for another contact. These 10 cells will clone themselves in order to multiply and then be able to fight. That's why we have this differentiation cloning of the cells, T and B, for the T cells. So the first challenge is we don't have as many T cells as we want. We can mobilize only a tiny fraction every time when we have new antigen. The second, different type of microbes require different type of immune response, of course. You can have intracellular microbes, you can have extracellular microbes, you can have sometimes in one time extracellular and you have to deal with this situation. MHC class 1 and MHC class 2. Here you have the MHC class 2, MHC class 1. MHC class 1 has a domain, what's called alpha chain, divided into 3 domains, alpha 1, alpha 2, and alpha 3. And it's, of course, it is a membrane, it is encrusted in the membrane, while it has also a beta 2 microglobulin, which is free. And of course, it has here this, what we call the possibility to take a peptide. The MHC class 2 is divided into 2 domains, alpha chain and beta chain. Both are, of course, into the membrane. And also it has the possibility to take a peptide. Both of them, they can carry this 10 to 20 amino acid peptide to be, of course, presented to the lymphocyte. MHC class 1. This molecule has a single binding site for a peptide. The site is created by 2 domains, alpha 1 and alpha 2. The receptor site is a form of a cradle, which can take, of course, only whose dimensions are largely sufficient to accommodate the peptide of about 10 to 20 amino acids, not more. That's why there are restrictions. Peptide and only 10 to 20 amino acids in this cradle, which can be carrying this peptide. The majority of variable amino acids, those that differ in the various allelic forms of the A and B or C molecules, are located in the bottom of the cradle where the peptide is to be bound. Function of the MHC class 1. Cellular proteins are degraded into peptide, which are attached to the class 1 molecule being synthesized in the endoplasmic reticulum. The whole is exposed at the level of the cell membrane. This set is recognized by the cytotoxic T lymphocyte as part of the cell. We will see the demonstration and then come back to the slide. Here we have a normal nucleated cell, healthy, not affected, but in a normal healthy cell, nucleated, any somatic cell of the skin, of the kidney, of the liver, we have some proteins, they need to be recycled. Either they are old or malfunctioning, or they need to be recycled. So all those proteins, they will go through an organ called proteasome, which will cut the protein into peptide, and peptide between 10 to 20 amino acids. This peptide, as you see here, will enter the endoplasmic reticulum through the peptide transporter. In the same time, in the meantime, the endoplasmic reticulum is where we can see the synthesis of what? The MHC class 1 molecule. This peptide will bind to this MHC class 1 molecule, and the whole is presented to the CD8 cell. Here we have the MHC class 1, we have the peptide. In our case, it's self-antigen, and then the MHC, the T cell will come with all the receptors. We see CD3, T cell receptor, which will look at this antigen if it is self or not self, and the CD8 to stabilize the connection between the cell and the T cell, CD8. We will go again and read. Cellular protein are degraded into peptide, which are attached to a class 1 molecule being synthesized in the endoplasmic reticulum. The whole is exported at the level of the cell membrane. This set is recognized by cytotoxic T lymphocyte as part of the cell. Why? Because it is cell protein. This cell is not infected. The second function is to present viral proteins or foreign antigen. Viral proteins synthesized by infected cells are also degraded into peptide, which are attached to class 1 molecules. The whole is recognized by cytotoxic T lymphocyte as being part of the non-cell foreign, which leads to the destruction of the cell target. Thus, the wide distribution of MHC class 1 molecule all recreated cell ensures in principle the protection of the organism against viral infection. Let's see the slide. Here we have an infection. Yesterday, the cell was not infected. The cell was just presenting only self-antigen. Today, the cell is infected by a virus, flu, cold, corona, any virus or bacteria. The virus, of course, tries to multiply itself inside the cell. But the cell will take some protein, viral protein, and take them through the proteasome as the normal function. In the proteasome, only immunogenic peptide go through and enter the endoplasmic reticulum through the peptide transporter. And in the peptide, in the reticulum, we will have the MHC class 1, both the immunogenic peptide, non-self, with the MHC class 1 cell will, of course, go through Golgi and then through exocytic vesicle, and the whole is presented to CD8. So basically, the CD8 cell is always facing two cases, two scenarios. In the majority of the case, the CD8 cell is facing healthy cells, which are presenting only self-antigen. This CD8 cell will not kill, of course, just will leave those cells having a normal life since they are not infected. In some cases, especially in winter or with this coronavirus, the cells are infected, not all of them, but the ones that are infected, they will present the foreign antigen, and the CD8 has to kill infected cells. So always in this situation, the infected cell has to die. Now, so the T cell is facing two situations. The first, on your right, is a normal healthy cell, which is presenting self-antigen. This cell will survive, the T cell will not harm, just let the cell living, and of course it will go to another cell controlling. That's why we are scanned every day. We are scanned by our T cells. Now, if the cell is infected, the T cell will make a stop, look at the GAN, If it is a foreign antigen, dangerous, the T cell will induce apoptosis. Why? Because if the T cell will not kill the affected cell, the virus will do it. And the virus will not kill through apoptosis, will kill through necrosis, which means it will make the cell explode, bigger and bigger holes, and the whole viruses, thousands of viruses will come out and infect more cells. So basically, it's who is making the first step. If our T cells are faster, healthier, they will kill the all infected cell and they will reduce the reservoir of the infection. Now, if our T cells are not healthy, the virus will make the step and make all the infected cells unfortunately dead through necrosis. So the T cell is facing four situations. Either the cell is healthy and the virus, the T cell will not kill the cell and will move to another healthy or unhealthy cells. Or the cell is infected, and here the T cell will kill the infected cells, which is the normal job. The third situation, in your right, the cell is healthy and the antigen is self-antigen, but unfortunately the T cell did not recognize this as self-antigen rather than a foreign antigen. With this doubt, the T cell can kill the normal healthy cell and then will make memory. Always we said T and B cells, they have memory because they belong to the acquired immune system. And what happened? What will happen here? Now, few T cells have killed those healthy cells, even if they have presented self-antigen. These T cells, they did a mistake. They will clone themselves and they will become 100 tomorrow and they will kill more healthy cells. And they will clone tomorrow again and they will kill thousands of our cells. And the autoimmune disease has started. But here when we have, unfortunately, autoimmune diseases. Autoimmune diseases are mainly the work of B and T cells. In our case here, T cells, because they did a mistake by mixing, not recognizing correctly the self-antigen and recognize it as foreign antigen. Why this situation is happening? Mainly, there is, of course, a genetic background, but our life, our hygiene life, our way of life, the stress, these T cells are so stressed, are so full with oxidative stress molecules that they cannot make the right decision. And unfortunately, they will, of course, kill healthy cells for almost forever, because that's what autoimmune diseases, and unfortunately, that's what is happening nowadays more and more. The fourth scenario, infected cell will present a foreign antigen, but CD8 will make either take too much time to take a decision or not recognize it as such, and then the virus will spread. So T cells, they have CD8 cells, they have a huge responsibility to make right decision in order to save our body and our organisms. MHC class 2. MHC class 2 are much easier. They are all only present on or expressed on monocyte macrophages, dendritic cells, longer haired cells, and B lymphocyte and some endothelial cells. Presented to whom? To CD4. So basically, these cells are professional, capturing antigen and presenting. These cells, or CD4 cells, they have, I will say, a little bit easier life. APCs can ingest microbes or microbial protein in different ways, either through binding, opsonization, phagocytosis, or endocytosis. Here it's professional cell that has captured the germ, as you see here, and and of course the germs through the phagolysosome is degraded, and you see here this immunogenic part, but there is no way that this germ or part of it will enter the endovascular reticulum. These cells are professional. They know that these end proteins are harmful. In the meantime, there is synthesis of MHC class 2. At the end, the MHC class 2 will bind to this foreign antigen and the whole is presented to CD4. So basically CD4 has almost one case, because here we ingest, we phagocyte only foreign antigen and we present only foreign antigen. But CD8 has almost four situations or four scenarios. CD4 cell, of course, will not kill the antigen presenting cell, rather will express, secrete cytokine that will recruit either other cells or, of course, induce immune response. That's what the job of CD4 cells. So the exogenous protein, which are in the endocyte by the antigen presenting cells, are also degraded into peptide in the endosome. Class 2 molecules also synthesized in the endoplasmic reticulum associated with the invariant gamma chain. This invariant chain masks the peptide receptor. The complex then migrates into the endosome, where the invariant chain associated or dissociated from class 2 molecule, the peptide can then bind to the whole, is exposed to the level of the cell membrane. The whole is recognized by the CD4 cells as part of non-self, which causes the activation of the onset of the immune reaction. So MHC class 2 only exposed or expressed on the surface of professional cells and presenting only a foreign antigen to CD4 cells. Degradation of antigenic protein, the extracellular protein that are retained inside the APC in the vesicles, are degraded and presented by MHC class 2 molecules. The intracellular protein of nucleated cells are degraded and presented by MHC class 1 molecules. And here you see a summary. MHC class 1, it is either self-protein or foreign protein, depends on the status, healthy versus infection. We have just immunogenic part presented, enter the endoplasmic reticulum through peptide transporter associated with MHC class 1, and the whole is presented to CD4 cells. CD8 cells. CD8 cells has to make a decision. If it is self, nothing happened. If it is a foreign antigen, has to kill infected cells and not to make a mistake. Now, MHC class 2, we have capture of the foreign antigen, micro, degradation, no entry to the reticulum, MHC class 2 synthesis. At the end, association between the MHC class 2 and the antigen, foreign antigen, and the whole is presented to CD4 cells, which will, of course, trigger the immune reaction. MHC class molecules, and MHC molecule present only one peptide each time. MHC molecules receive their peptide during the synthesis and assembly within the cell. MHC1 receive the peptide that are found in the cytoplasm. MHC2 receive proteins that are found inside intracellular vesicle. In each individual, MHC molecules present foreign peptide that belong to microbes, as well as proteins specific to the individual. This inability of MHC molecule to differentiate between self and non-self antigen raises two questions. Yes, indeed. Even if the cell is infected, not all MHC molecules will carry the foreign antigen. No, some of them still continue to present self, and some of them will present the non-self. And in this situation, we have two questions. Question one, it is accepted that at each moment, the protein of the self are in much greater quantity than the microbial protein, of course. Why don't self protein constantly occupy a MHC molecule? So how come that few of the foreign molecule can have a space and a place to be presented to the T cells, especially CD8 cells? The answer, the new MHC molecules are always constantly being synthesized and are ready to capture every peptide found in the cytoplasm. It's like trucks in a factory. The truck comes, it will be full loaded and move. And MHC class 1 cells, they are always carrying what entered the reticulum, either foreign antigen or self-antigen. A T cell needs 0.1 to 1% of an average of 10 to the 5th MHC present on APC. The very rare microbial molecule presented by MHC are sufficient. And here the extreme professionalism, the extreme affinity of T cells to foreign antigen. If you have 1% of MHC out of 100,000 carrying foreign antigen, the T cell CD8 cell can recognize this 1%. That's what makes these T cells extremely competent and extremely professional. The second question, if MHC molecule constantly present protein of the cell, how do we not develop immune responses against our own antigens? How we are not making the third scenario, when the cell is present in the self and the T cell is not making a mistake, autoimmune response. The answer, the cell that can respond to self-antigen are either killed or inactivated, and it is what we call the thymic selection or negative selection that takes place in the thymus. And that's what we will see, of course, in the next session. Thank you very much for your attention. I hope you enjoyed, and we will complete the story in the next session. Bye.